Hello friends, welcome back to NitVed Bioscience. And today's topic of the discussion is the quaternary structure of the protein. Quaternary structures. In the previous few classes, we had discussed about the primary, secondary and the tertiary level. And today we will discuss about the quaternary structure of the proteins. So first we will understand what is the quaternary structure of the proteins. So quaternary structure are the proteins which is having the quaternary structures are those protein in which more than one polypeptide chain are present, and there is some sort of the interactions or the bonding between this individual polypeptide chain, and together they will carry out some biological functions. So that is known as a quaternary structure. Individual polypeptide chain is known as a subunit. Okay, all protein will have the primary, secondary and the tertiary levels. Okay, all the proteins will have the primary, secondary and the tertiary levels, and those proteins are the functional at the tertiary levels. But in some proteins, more than one polypeptide chain, here only the single polypeptide chain is present, here the more than the one polypeptide chain is present, and they work in a association. So this is a quaternary structure. So this is how the quaternary structure is different. So not all protein will have the quaternary structures, but all protein will have the primary, secondary and the tertiary structures. For quaternary structures, more than one polypeptide chain needs to come close to each other. So that is the subunit. So individual subunit, each individual subunit will have its own primary and secondary and the tertiary level. Okay, subunit. Now if two subunits are present, then it is known as a dimer. 3, then trimer, and likewise dimer, trimer, tetramer, pentamer, depending upon the number of the subunit is present, means the quaternary structure is having the oligomeric protein. Got it? So this is the introduction of the quaternary structure of the proteins. Now there can be the chances that this individual subunits, they can be identical or can be the different. Okay, it can be the same or if it is a difference. So if it is the same, then it is known as a homomeric protein, and if it is a different, then it is known as a heteromeric protein. Homomeric protein and heteromeric protein. What is homomeric protein? In the homomeric proteins, all the subunits are same. All subunits are same or identical, and they will synthesize from the same gene. Identical subunit synthesize from the same gene. Okay, and the example is a lactate dehydrogenase or beta galactosidase. And what is a heteromeric proteins? In the heteromeric protein, the subunits, they can be the different subunits are present, which will synthesize from the different genes. And the example, most common example is a hemoglobin. Hemoglobin, it is made up of the 4 polypeptide chains. So out of the 4, 2 alpha chain, 2 beta chains. So this is the example of the heteromeric proteins, because all the 4 polypeptide chain are not identical. So here the example is hemoglobin. Okay. So what we had discussed, first we had to understand what is the quaternary structures, then the subunits, if the 2, so dimer, trimer, tetramer, and if it is identical then it is a homomeric protein, if it is not identical then it is a heteromeric proteins. Now we will see the example of the quaternary structure of the protein, which is mostly involved in the biological process. So hemoglobin, DNA polymerase, ribosome, okay, antibodies, ion channels, these are the some example which is having the quaternary structure of the protein. And what is the function of the quaternary structure of the protein? So most common example of the function of quaternary structure is like a gene expression, macromolecular transport, okay, various chemical reactions and the regulations. So for all this activity, the quaternary structure of the protein is required. Now the next we will see what are the methods to identify the quaternary structure of the protein. So the most common one is the X-ray crystallography method. Through X-ray crystallography we can identify the structure is having the quaternary structure or not. If there is any difficulty to obtain the data from the X-ray crystallography, then the NMR or electron microscopy can also give the clues about the quaternary structures, and the molecular weight of the each subunit can be obtained by the size expulsion chromatography. There are also there is a list of the method, but I have mentioned here only the few of them. Now these subunits of the quaternary structures must be specifically arranged for this entire protein to function properly. Subunit must be specifically arranged. So if there is any changes in either the structure of subunit, individual subunit, or how they are interacted, so it will make the marked difference on the biological activity. So now we will see the what kind of the interactions or the bonds are present which is holding the quaternary structures or which is holding the all the subunit together. So together they will perform the biological activity. So those interactions we can divide into two categories: non-covalent interactions and the covalent interactions. Non-covalent and the covalent interaction. So the interactions or the bonding which is holding the quaternary structures or the subunit of the quaternary structures, this is mostly of a non-covalent of the types. So the interactions can be the hydrogen bond, hydrophobic interaction, okay, van der Waals interaction, and electrostatic interaction, which is also known as a ionic bond. These are the list of the mostly the non-covalent interactions which is holding the subunit of proteins. And for the covalent interactions, these are the sulfur bond or the sulfur bridges, disulfide bond, okay, more specifically we will write here disulfide bond. So this is about the bondings. Now we will discuss about how the subunits of the quaternary structure will carry out its biological activities. So there can be of two types: cooperative effect or allosteric regulations. For the cooperative effect, we will take the example of hemoglobin. Just we had discussed that the hemoglobin has a 4 polypeptide chains, 2 alpha and the 2 beta chains. And what is the main function of hemoglobin? Hemoglobin main function is the transport of oxygen. So transport of oxygen, what happens here? If oxygen molecule will bind to any of the subunit, if oxygen will bind to any of the subunit, there will be some conformational changes of the other subunit, so that the affinity of the other subunits towards oxygen will increase and the other oxygen will also bind there. Okay, binding of oxygen to one of the subunit will make conformational changes of the other subunit, so that the affinity of the other subunits toward oxygen will increase and the other subunit will also bind to the oxygen, and so on. So this will have the effect here. Then the affinity of the other subunit will increase and oxygen will bind, and finally the fourth. So that is how the binding of the oxygen to one subunit will have the effect on the others. Means affinity of the hemoglobin towards oxygen will increase and the more oxygen molecule will bind here. So that is a kind of the this is the cooperative binding or the cooperative effect. Here only the one side is involved, that is the active side. Next is the allosteric kind of the regulations, which we will mostly see in the enzyme substrate reactions, enzyme substrate chemical reactions, or receptor ligand which is involved in the cell signaling. We will see the allosteric regulation. Allosteric regulation, for the allosteric regulation, let's take the example of some dimeric proteins. There can be the more than one polypeptide chains, but we will see here the dimeric proteins. In the here there are the two type of the sites are present. One is the allosteric site and another is the active site. And this allosteric regulations can be of two types. It is a positive regulations or the negative regulations. Positive regulation, as the name suggests, it will increase the activity if it is an enzyme or in the cell signaling, and negative regulator will, it will stop or decrease the activity. So if we consider this is any of enzyme, okay, if positive regulator, this is positive regulator. Positive regulator will bind here, so that will make the conformational changes so that any substrate will bind to enzymes and it will carry out its functions. That is a positive effect. In case of the negative regulator, same way, negative regulator will bind here, okay, conformational changes, so that the enzyme substrate reaction will not take place and it will stop that activity. Same with the cell signaling. Cell signaling, either we want to increase the rate or we need to start the cell signaling. So in that case, the positive regulations, and likewise that negative regulator will bind and the cell signaling process will stop here. So this is the brief idea about the quaternary structure of the proteins. Okay, quaternary structure has a more than the one polypeptide chains. It can be homomeric or heteromeric. Homomeric proteins are heteromeric. Individual is known as a subunit. There are some method to identify the quaternary structure of the proteins, and these are the two ways by which the quaternary structure functions, that is a cooperative binding or allosteric binding, and the important, the interactions. Interaction, mostly it is a non-covalent type of interactions, and some covalent interactions are also present. I hope this video is useful to you. If you are finding it useful, please like, share and subscribe. Thank you.